I'd like to welcome you all here tonight. Thank you for joining us at our very first rama lecture called Illuminating the Invisible, New Developments in Psychiatric Genetics and Their Potential for Clinical Use. I'm Professor Tracy Wade and I'm the Director of the rama Institute for Mental Health and Wellbeing, and I'm delighted to be hosting this event tonight. First, I'd like to acknowledge that we're hosting this forum on the traditional lands of the Kaurna people. We pay our respect to the elders past, present and emerging. We acknowledge and convey our deep appreciation to the elders of all the nations where Flinders has its presence. Now, it's really wonderful to see you here today. It's particularly wonderful because of the whole COVID thing, that we can get together at a slightly distant sort of way. And I'd like to take the opportunity to acknowledge the Honourable Stephen Wade, MLC, Minister for Health and Wellbeing. I'd also like to acknowledge Professor Mike Kyrgios, who's the Vice President and Executive Dean of the Flinders University College of Education, Psychology and Social Work. And we also have the rama Advisory Board member, John Mannion, who's just put food in his mouth. John has multiple hats. He's also one of the mental health commissioners, and he's also the breakthrough guy. And Breakthrough has been a very great ally for rama and for funding research, and so it's a very special relationship. Now, I have to tell you about, I'm going to do my air hostess, the emergency exits located to my left, through the glass sliding door towards the stairwell there. The meeting point is Victoria Square. Restrooms can be accessed through that glass door and turn left and go down the hallway. And tonight's event is being live streamed, so recording will be made available to all who've registered in the first 24 hours. For the people that are viewing online, please feel free to submit questions via Slido using the code hashtag rama2021. And the questions will be sent to me and we will ask Nick in the question time. Also, feel free to join the conversation on Twitter using the hashtag ramalecture2021. Now, we do ask that everyone please treat this forum as a place of respectful engagement, where people are treated with dignity and where differing views are tolerated. Before introducing tonight's keynote speaker, Professor Nick Martin, I'd first like to invite the Honourable Stephen Wade, Minister for Health and Wellbeing, to say a few words. Welcome, Stephen. Thank you for joining us tonight.
Thanks very much. I'd like to acknowledge Professor Tracy Wade, Heather Holmes-Ross, the Mayor of City of Mitcham, John Mannion, the SA Mental Health Commissioner and rama Advisory Board Member, Professor Nick Martin, Senior Scientist and Senior Principal Research Fellow at QIMR Beckhofer Medical Research Institute, Sigira Nandu, the Chief Executive Officer of Sonda and the rama Advisory Board Member, and Professor Sharon Lawn, rama Advisory Board Member. I too would like to join Tracy in acknowledging that the land that we meet on today is the traditional lands of the Kaurna people, and I pay my respects to their elders past, present and future. I get a lot of invitations to events in my work. Most of them I decline. This one I couldn't. It's a pleasure to be part of this, the Inaugural rama Institute for Mental Health and Wellbeing Inaugural Lecture on Illuminating the Invisible, New Developments in Psychiatric Genetics and Their Potential for Clinical Use. The advent of the global pandemic and the unprecedented development of a suite of vaccines has highlighted the transformative power of health and medical research. The pandemic has also turned the spotlight on mental health. Faced with the trauma of mass deaths, the social isolation of lockdowns and the stress of uncertainty, COVID-19 is challenging the well-being of communities right around the world, including their mental health. But as we spend billions of dollars on vaccine research to deal with the physical impacts of the virus, we don't see a proportionate investment in our mental well-being. In early 2019, before the pandemic, Professor Pat McGorry launched the rama Institute. Flinders University wanted to bring together the university's expertise in both mental health and wellbeing. It'd be fair to say that I've taken a personal interest in the journey of Orma. The director is the most talented member of the household of which I'm a part. During the COVID-19 pandemic, Orma Institute partnered with the Samri Wellbeing and Resilience Centre to deliver a new program called the Be Well Plan, a program that helps craft a unique evidence-based strategy matched to people's needs and unique circumstances so they can stay feeling well and grounded. This is a terrific example of the way that research informs and leads to pragmatic and useful outcomes and tools for communities. It's 592 days since the first positive COVID case in South Australia. But the pandemic is far from over. The Black Death lasted 7 years. But what is clear is that no matter how long the infectious diseases challenge remains, the impact of the pandemic on the mental health and well-being of our community will be long-lasting. Mental health and psychiatric care and research and development in South Australia has never been more important. As mental health services seek to attract and retain the best personnel in the face of critical workforce shortages in health professions, the State Government's Mental Health Services Plan 2020-2025 promotes academic research and development activities. The opportunity to be involved in research and development within a chosen field is a strong factor in retaining staff through providing a meaningful and engaging activity that promotes career development and contributes to a broader knowledge base. Research into the genetic basis of psychiatric disorders is a rapidly expanding area, and that has and will provide powerful insights into the biology and precision medical management of these disorders. Professor Nick Martin and others have, through their work, demonstrated that major psychiatric disorders have a familial and heritable component. Research such as this allows us to better understand the role of genetics in mental illness. This work may ultimately lead to the identification of those most at risk, early intervention, the development of more targeted treatments and therapies, and optimal early management of a range of conditions. So I'd like to thank Professor Martin for running the gauntlet of border closures to be with us today, for isolating until he had his first test, and for observing sound public health practices. In concluding, I'd thank Orma Institute for Mental Health and Wellbeing for the work that Orma is doing to promote the health and wellbeing of South Australians, and wish you all the best for your future endeavours.
So, without further ado, I would like to welcome Professor Nick Martin. We're very lucky to have him here. He's a renowned geneticist, senior scientist, senior principal research fellow at the QO Meyerberg Hoffer Medical Research Institute. Nick Martin graduated with honours from our own University of Adelaide here in genetics in 1972, just yesterday, really, wasn't it? And he obtained his PhD in genetics at the University of Birmingham, that's in the UK. In 1978, he returned to a research fellowship at the Australian National University, where he founded the Australian Twin Registry. After 3 years in the US, he returned in 1986 to the Queensland Institute of Medical Research, where he heads up the Genetic Epidemiology Laboratory. And he continues longitudinal studies with twins that have a wide range of complex traits of both medical and behavioural interest. He's also involved in several large studies of cognition and brain imaging using EEG and MRI. His research over recent years has moved to genome-wide association studies, or GWAS as we call them, to locate genes influencing complex traits, including anxiety, alcoholism, and dizygotic twinning. He's developed methods for multivariate analysis and analysis of gene-environment interactions. More recently, he's initiated projects to recruit large patient samples for GWAS of anorexia nervosa, depression, and other psychiatric disorders. He's published over 1,400 papers in peer-reviewed journals. He's a fellow of the Australian Academies of Science, Social Science, and Health and Medical Science. That's 3 different academies, that's just not one academy. So it's my pleasure to welcome Nick to deliver this evening's keynote, Illuminating the Invisible, New Developments in Psychiatric Genetics and Their Potential for Clinical Use. Thank you, Nick.
Thank you very much, Tracy, for that warm welcome. Minister, for being here. It's a great pleasure to be back in Adelaide, my original home, as Tracy pointed out. I'm sixth generation Adelaide, lived my first 23 years of my life here, and was forced away by a better offer to Brisbane, where I've been ever since. But it's great to be back here on this absolutely gorgeous spring day and to be giving this inaugural lecture. And if I can find my slides. So my topic is psychiatric genetics, which I'd love to have a whole disquisition on the origin of this subject, but I mean, just to show in one slide, I think we can credit the Germans in the first part of the last century, and actually the late 19th century, but of whom the most eminent was Emil Kraepelin, who was the person who originally described what we now know as schizophrenia. He called it dementia praecox, and was the person who actually realised that this often ran in families, and promoted the systematic study of that, including of twin studies, which provided the evidence that in fact schizophrenia and many other mental conditions are influenced by genetic factors. So how do twin studies work? Well, they depend upon the existence of these two types of twins: identical twins who share all their genes in common, and you can see that in this slide here of a profiler genotyping. This is the sort they use for criminal investigations. And you can see this pair of identical twins here share all of their genes in common, compared with non-identical twins who are just like ordinary brothers and sisters, except that they are conceived, gestated and born at the same time. So they form the perfect control for MZ twins in the circumstances of their birth and their upbringing. And DZ twins on average share about half their genes, and you can actually see here for these twins, some of the bands are the same and some are different between the twins, and on average it's about 50%. So with a little bit of algebra we can compare the similarity of identical and non-identical twins to calculate the proportion of variance due to genetic factors. So once people realise the power of this study, they really took off, and these are results of studies of a whole range of complex conditions, of which the first are 4 psychiatric and the last 4 are not organic. But you can see in the black bars the concordance of identical twins versus, in the grey bars, dizygotic or non-identical pairs. Concordance means if one twin is affected, what proportion of their co-twins are affected, and what's the chance that their co-twin can be affected. And you can see that for all of these complex traits, well, with the possible exception of MDD, major depressive disorder, well, in all cases, the MZ twins are more likely to be both concordant. And so you can actually do a bit of algebra to then estimate the heritability. That's the proportion of all the variation in these traits due to genetic factors, and there are hundreds of such studies, but just summarised here in round numbers on this study there. I mean, most remarkably, we see schizophrenia not on there as ADHD, for example, which is also about 80%, extremely genetic. Autism right up there, about 80 or 90%. Most people think bipolar is a bit lower at around 60%. Depression, everyone now agrees after many, many studies. I think the last estimate was 37% from all of the studies, and you can see contributions there for the anxiety and panic disorders, and also we're not going to mention otherwise into substance abuse, including alcoholism, which is about 60%. So these are all the estimates from twin studies, and of special interest here, since I'm in Adelaide, where Tracy has pioneered studies of anorexia, and in fact I did have some role in her PhD too, which was rather nice. So these were studies published by Tracy and several other groups of twins around the world, all of which were homing in on an estimate of around 60% heritability. And in fact, in another study that she and I co-authored. We also found this magic number of 60% for behaviours around various eating disorders, not just anorexia. So strong genetic component there. So these are the things we know about anorexia, and I think many people in this room will already know them, but the big question is, how do we find the genes? And I might say that that's more or less where the subject stopped up till about 2005. And basically people like me and hundreds of others around the world wasted. We didn't waste, but we spent a lot of time and a huge amount of money that went nowhere in the 80s and 90s and early parts of the noughties, really not getting very far. And then suddenly came along the realization that the only way we're going to find these genes is with what are called association studies, where you're looking for association, just simple statistical association between a genetic marker and your disease. And the genetic markers we use are what we call SNPs, which is short for single nucleotide polymorphisms. So these are strands of DNA with the 4 letters that I hope you all know, C, T, A, and G, and you know, arranged 3.3 billion of them in the human genome. And about every 1,300, you get a site that's variable between people. And that's called a polymorphism. Our Greek director over here will know perfectly well what that means. It means many forms. In this case, there are just 2 forms, but sometimes you can get more than that. So we home in on using these SNPs. And what really caused the breakthrough was our ability to type these economically spread right across the genome. So this is just a schematic showing that what we're trying to do is to look at, you know, everywhere across the genome. And in fact, the density of SNPs we have across all of the 23 human chromosomes is actually much higher than this. It's just really a cartoon. And what made this possible was the breakthrough about 2005. In the discovery of, the engineering, it's the most remarkable piece of engineering story that hasn't really been told very well. It's a great book or 2 in there, I think, on the development of these chips, which started off as individual chips about the size of a postage stamp on which you could type about 100,000 SNPs. And now we're at the point where on this slide, which is about the size of a regular microscope slide, we've now got 24 miniaturized such chips. Each one of those can type about 1 million SNPs. So we can type 24 people on one chip like that. And that is, when the first chips came out in 2005, they were in the dollars of the day, about $2,000 a pop. Now you can buy a chip like that so that the unit price is under 20. So, and that's what really has made this work that I'm going to talk about possible. It's just that incredible development of technology and driving down the cost. And that's been really important because as time has gone on, it's become evident that the studies we need to do involve typing huge numbers of people. So just to hammer home the way we do these association studies, these genome-wide association studies, or GWAS, as Tracy mentioned, is we have a whole bunch of cases and we have a whole bunch of controls, and we type them on these chips. And then we do a simple, for every SNP there, we do a simple association test. It's just a simple 2x2 chi-square test. Statistics 101, lecture 1, you know, really simple stuff. And you say, is this SNP associated with my disease? And this is what's called a Manhattan plot for obvious reasons. And along the X chromosome here, you can see all of the chromosomes going from 1 up to the X chromosome over here. And up the y-axis is the strength of association for association with that disease. And what you're looking for are these towers, these Manhattan tower blocks, which says, bingo, there's a gene here, or very, very close to here, which is affecting my disease. And so I just love this stuff. I think it's really exciting. The only problem is, because you're doing millions of tests, you've got to really be careful about getting results by chance. So what we have to use is what's called Bonferroni's correction, whereby we correct that 0.05 statistical level, 1 in 20, that's the one you remember from Stats 101, but we have to divide that by 1,000,000 to get the right level, which means that we have to pass this bar here, which is 5 by 10 to the minus 8. So it's an extremely stringent criterion for significance to find these. All right, so this is what we have done with anorexia, and this is a consortium of ourselves here in Australia. We contributed to these global 17,000 cases. We actually contributed 3,000 of those, which we're rather proud of, and then more from the UK, from the US and Sweden and Denmark for this total of 17,000 cases, 52,000 controls. And doing our GWAS, this is the first GWAS in which we actually found hits, we call these hits, for anorexia. And we've got 8 such hits. These sort of rather surprised everybody, because everybody expected to find hits that we'd previously found associated with psychiatric diseases. And indeed some of these are, but more than half of them are actually involved in metabolism and body size, and also in sundry other diseases. So that was really a big surprise as to what that means. It's not just, we weren't just finding these neuropsychiatric genes, we're finding metabolic and genes involved in autoimmunity. And so on the one hand, one is interested in the individual peaks, but the other thing you can do with GWAS results is calculate what's called a polygenic risk score. And so for every SNP going right across the whole genome, we can then ask, well, what is the effect size of that SNP according to that association test we've done? And we can then multiply that by the frequency, by whether that person has that SNP or not. And for every individual, we can calculate what's called a polygenic risk score. And that obviously is going to be different for every disease that you do it, and for every individual. And it's turning out that, in fact, it's that polygenic risk score which is really, in many ways, more useful than the individual SNP results, at least at this stage. I'll show you how that transitions. But right now we're very excited about PRS. And just to show you one of the best stories around, this is schizophrenia, where, which was really the main focus of psychiatric genetics. It's always been the premier disease, so most effort and money has gone into it. And this is the results of the polygenic, of the GWAS for schizophrenia based upon about 30,000 cases, 40,000 controls. And using the results from that to then predict into a new totally independent sample of about 11,000 cases from the UK. And so the estimation of your prediction function is completely independent of the group that you're predicting into. That's the key for this sort of work. And what it's showing is that polygenic risk score, if you're in the top 1%, so these are the polygenic risk scores of the whole sample in this new prediction sample of 11,000. The polygenic risk scores of all of those people arranged in descending order from here. And what it's showing is that if you're in the top 1% of those polygenic risk scores, you're at 40-fold higher risk of getting schizophrenia than if you're in the bottom 1%. And as I've sort of said rather coyly over there, I think at that stage it starts to become politically useful. And the question mark there is if you can sort out the ethics and how you're going to do it, because it does raise many practical ethical questions. But leaving those aside, from a purely scientific point of view, this starts to become interesting. We can talk about ethics later, if you want to. And so now coming back to anorexia, where did our anorexia GWAS get us to? So this is actually taking that GWAS that we had for anorexia, but now it's taking out Australia and New Zealand, and just using the other countries, US, Sweden, Denmark, and saying, well, how well do we go predicting from that back into Australia and New Zealand? And the answer is, it's not nearly as dramatic as schizophrenia, but it's not bad. I mean, already, you know, looking in the top decile here, the top 10% of polygenic risk scores have a chance of getting anorexia about threefold higher than if you're in the bottom 10%. So, I mean, you could imagine sometime in the future, if ever we can sort out the ethics, I mean, you know, you might have prospective screening of people. I don't know, maybe this is going to be done at birth. If you've seen that film Gattaca, you know, it's sort of presages this sort of thing. And you could actually tell parents, well, you know, your child has got a very high risk, genetic risk of becoming this. You should watch out for it. You should make sure that they're not exposed to too many fashion magazines or something. I don't know. But we could speculate about that, the sort of use that it might be put to. I mean, at the moment, this isn't very spectacular. The only thing I can predict with absolute certainty is that that's going to get better and better. You know, with the new studies we do, as the sample sizes go up, that's just going to get more and more powerful. The other thing we can do with polygenic risk scores is calculate what are called genetic correlations. And so we're doing GWAS not just for anorexia or schizophrenia or whatever, we're doing it for every trait under the sun, all these ones that are listed on here. In fact, I think GWAS has been done now. We've got really good data on at least 2,000 traits, and that includes diseases, yes, no. It includes quantitative things like cholesterol level. It includes a lot of psychological measures, IQ, body mass index, and so on and so on and so on. And so this is just looking at the correlations, the top correlations of the anorexia. So for every one of those, we have a vector of loadings on all the SNPs in a GWAS array, okay? And they've all got their little measure. And then you take your second trait, say it's BMI, and that's also got a GWAS for all the same SNPs. So you've got this whole thing. And you just do a correlation of one with the other. So it's quite simple. You're just correlating one set of weights with the other set of weights, and that gives you a genetic correlation. And these can be from different samples. They don't have to be done in the same sample. The constant reference point always is that SNP chip. We always use the same chip. And so we get these genetic correlations out. And so if there's nothing going on at all, they're going to be zero on this line here. But what we're finding is a bunch of these correlations which are positive and some others that are negative. And what we're finding positive is, not surprisingly, correlations with psychiatric disorders. And guess which the highest correlation is of all? With obsessive compulsive disorder. Now, Tracy could have told you that already, but it's really neat to actually see that result confirmed in this completely independent way. And that's a genetic correlation of 0.5, which is pretty high actually, but you're also seeing correlations with, not surprisingly, with depression, with schizophrenia, anxiety, with the personality trait neuroticism. You're seeing correlations with positive correlations with education, college completion. We know that. We know that anorexics tend to be more highly educated. And we also know that it's positive with physical activity. We know they exercise more. So those things all make sense. What was more surprising is finding these quite strong negative correlations with all these diabetogenic traits here, and even stronger, surprising yes or no, that we find these strong correlations with body size parameters here, because you know, the assumption is that that anorexia, the body size perturbations, are a result of the disease. But what this is saying is that these people already have got an underlying perturbation of their body size profile. So, you know, quite what that means, how to interpret it, you know, we can speculate on. We can just say it's just work in progress, but it's pretty interesting. So where do we go from here? I think we need to do a lot more work on this association with metabolic mechanisms and so on. But of course, immediately you start to then wonder, what about the other eating disorders? In particular, bulimia and binge eating. And in fact, we've just launched a new project called EDGI, which stands for Eating Disorders Genetics Initiative. This is funded by the NIH. and in which we're recruiting people not only with anorexia but also with bulimia and binge eating, and they do an online survey, and if they meet criteria, then we get a saliva sample from them. We've got a really good pipeline now, as it were, for collecting saliva samples from people and extracting DNA from those, sort of trying to minimise the cost. We've got the cost down to about $30, which is really good. So now I want to talk about the work we've done on depression. I mentioned depression. This is the most prevalent of the psychiatric conditions, up to 20% of females, half that number of males, and that ratio there is just a universal constant, and no one has got any good explanation as to why. So the heritability here is considerably lower than for anorexia. So for GWAS, your ideal is a trait that's got really high heritability and really low prevalence. And that's why schizophrenia has been so successful, because it's got a 1% prevalence, 80% heritability. That's why we get such huge power with samples of only 30,000 cases. But when we come to something like depression, where you've got a heritability half that, but 20% of the population have got it, or the female population anyway, you're really pushing uphill. So that means we're going to need huge numbers, and so it's proved. In fact, a colleague of mine calculated we need at least 5 times the sample sizes we needed for schizophrenia, and so it's proved. So this was the first, we had about 3 or 4 goes at this, and found almost nothing, and this is the first article that was only just quite recent, 2019, with 250,000 depression cases and double that number of controls. And at last we've got some findings that look pretty interesting. We've got 102 genes here. Schizophrenia, I think we're up to about 500 or something like that. So it starts to look good. But we can do the calculation, and I can't go into the statistics here, but a rather brilliant PhD student did the calculation for all of these different traits of what proportion of the genetic correlation, of the genes that we're looking for, we would expect to find given the sample sizes across here. These sample sizes are log 10. So that's 1,000, 10,000, 100,000, a million, 10 million, 100 million. And you can and these traits range from C-reactive protein, triglyceride, all these sorts of things, volume of the putamen in the brain, total cholesterol, blah, 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 blah. Here's schizophrenia, the blue one. And down here we've got some personality traits, and somewhere in here, here's depression, this light blue. And what it's showing is that for something like C-reactive protein or triglyceride, even with a sample of say 50,000 people, we've already got 80% of the genetic variants. So really strong statistical power. For the best of the psychiatric conditions, namely schizophrenia here, with the sample size that we've got, which is, this was actually a slightly later slide than the earlier one, that's up around 40, 50,000 people. We're starting to get to about 50% of the genetic variation. For depression over here, even with that, the study I just showed you, we're down here at about 3%. And that was the estimate we actually got of the proportion of variation that we've accounted for. And we can predict that to actually get up to 80%, we're going to need somewhere between 10 million and 100 million people. So we need ways of recruiting people really efficiently. We can't sit around waiting for a psychiatrist to do 3-hour interviews. We've got to have some smart way of doing it. So I had the thought of 2 smart ways to do it. It was one, to make use of the pharmaceutical benefit scheme and actually define the disease by the medications people take. So we actually asked the PBS to mail out to people who had at least 4 prescriptions for the top 10 antidepressants in Australia and asked them to take part. Well, that was partly successful. It's been more successful in our study of bipolar, where we've just been targeting people who use lithium. But for depression, it wasn't particularly. So the other way was just pizzazz. Let's just do a PR campaign. So we hired a top medical PR company in Sydney to design a sort of media blitz, and we ran that over a couple of days, and we recruited over 30,000 patients in about a week. And those are then invited to go to our website, where they do this fairly comprehensive online survey. There's a core survey that all around depression and the medications they're taking for it and the side effects and so on. But we also ask in all these domains here about all the things that are associated with depression. And my guess is that these encompass all the research interests of the people in this room, from a bit of chatting I was doing before. I think we've got PTSD and trauma and everything's in there. So if you want data on lots of people, come and see me. And of course, the other thing you need for these studies is controls. And we were lucky in having access to a control study from our institute who had screened for depression, so we could do that and get them GWAS. Now, this is what the distribution looks like of polygenic risk scores in your cases. That's blue, and control. So all you're doing is shifting the distribution a little bit to the right. But if those of you who know who've played in this space, you've only got to do that, and you vastly alter the proportion of people in the tails in getting a lot more cases. And these polygenic risk scores predict very well measures of severity, including the number of episodes people have had, their age of onset. These are the people who have early onset, late onset, and these are the people who are currently depressed versus no depression. So they've got really good value. And then by adding our results to that early analyses, what we've now done is to increase the sample, along with those other studies, to over 500,000, and we've now got over 500 genes for depression. And without our study, there would have been 50 odd fewer, which is sort of rather pleasing. So I don't have time for that. I'm running out of time. We're doing a lot of work on response to antidepressants too, with the aim of trying to work out who will respond to which drug, which would save a huge amount of health dollars. I haven't got time to do that. I just want to tell you one last story, and that which the Minister I know will be interested in, because where's this going? What about new treatments? What about new drugs? Well, so what we're trying to, the thing is that GWAS is great, but the SNPs that we're finding are common SNPs, and they tend to be of rather small effect, so they're not of particular interest to drug companies. What drug companies want is an effect that is huge. They want to see a really big, huge, and they want to know that that person who's carrying that gene is still alive. In other words, that having that genetic knockout, as it were, does not kill the patient. Because if you can find such variants, then it tells you could design a drug against that gene, against that protein, and knock it out with a pharmaceutical, and the patient won't die, but you'll have a dramatic effect on the disease. So this is what's been put into place, and this was a paper published only 2 months ago in Science, and I'm proud to say that one of my other PhD students was senior author on this, which involved using a technique called exome sequencing on over 640,000 people. And the trait they were interested in was body mass index. And so these are the results, and slightly complicated. So what you have here, up and down here, is the effect size, and here's 0, which means there's no effect at all, and in that direction is positive effect size, and here you're decreasing BMI. So above the increasing BMI here, you're decreasing. And then going along the x-axis here is the frequency of that variant. Once more, it's on a log scale, so these have got a frequency of about 10%, these are about 1%, these are about 1,000th, and so on. And by typing that huge sample, they were able to identify this variant in a gene called GPR75, which results in lowering body weight in heterozygotes. That's mean those are the ones who carry only one copy, and that's already lowering it by 5.3 kilograms. So if you're a homozygote, if you could completely knock that gene out, you would actually lower by 10 kilograms. So, and it turns out this class of drug, they're called G-protein coupled receptors, are the absolute ideal drug targets. These are the ones that the pharmaceutical companies dream about. And so this company, Regeneron in New York, is now going hell for that. They are spending billions of dollars aiming a drug like that, aiming to find a drug for that. This all in the last couple of months, this is happening. They're just going absolutely ape, because you can imagine what a drug like that would be worth on the worldwide market with the obesity epidemic, if you could actually get a drug that was harmless apart from knocking your weight right down. So that's the golden aim. And so my conclusion, and I realise I'm running late, as always. Just to prove their point, they then did what's called a mouse knockout experiment, and they actually took that gene and they knocked it out, and they did crosses, and they showed that if you're carrying one copy of the gene, in those mice, you're decreasing their weight by 25%. If you're carrying two copies, which is what we call a knockout, by 44%. So, if you believe in mouse studies, it's pretty exciting. And so my concluding remark is, I think this is fantastic. This all grows out of GWAS. I should have mentioned that we're looking that most of these dramatic, huge effects occur in genes that we've already found by GWAS as having much smaller variants, so more common variants, but of small effects. And so we know that that's the gene that's suspect. If we can then find in that gene these dramatic, huge effects, these are things that we can design drugs. And this is what we need to do in psychiatry. And what that means is we're just going to need to find these huge numbers of cases and sequence them. And Regeneron are now sequencing 1 million people a year. They'll sequence samples from anywhere. I'm sending all my samples there. And the IP we can sort out later. I don't care about that. The main thing is to get new drugs onto the market. Regeneron, by the way, have got a research expenditure this year of $2.8 billion US. That's for a research group of about the size of 150. That's more than the total of NHMRC, ARC and MRFF, this one company. But that's the future, I think. All right, I've spoken long enough. Thanks.
So I'd like to thank Nick very much for a very energetic and interesting and provocative talk. And we do have time now for questions. So we might get some online questions, but if you want to ask a question here, it'd be great if you could just come up to the mic. Try not to touch it or lick it, but just, just talk into it. But Nick, I did want to just go back. You sort of did allude to the ethical issue about testing people. I'm sort of, because I think I know what ethics committees think about these things, which is, really that it's all very difficult. But I'm wondering, do you have a better feel for what the ordinary person thinks? Because you've had so many years with so many different disorders. Do you get a sense of how ordinary people feel about
Well, I'm a libertarian. I'm a great believer in people making their own decisions, if they're properly informed.
Yeah.
And I think people should be given the choice as to what they want to do. But now, here it becomes a bit more awkward because you're making decisions for minors potentially, so it's got those sorts of issues. But if you look at adult behaviour and you look at these direct-to-consumer companies like 23andMe, which I've enthusiastically taken part in and paid my $100, $200 or what it is, and they will send you back, everybody gets their report on their ancestry, and everyone's interested in that. Well, most people are, and most often it confirms what they already knew, but sometimes there are more interesting nuances that come up, and sometimes some surprises. But what most people don't know is that when you get that report back, you can actually go around their website and there's a place where you can press to download your own genotype file. You can take that genotype file to another free company called impute.me and upload it. And a couple of days later, you get back a report on 500 diseases arranged in alphabetical order with your percentile risk on all of these. And in fact, the person, my colleague Naomi Ray in Brisbane, who's the person who developed all this theory and applied it, another Australian triumph, by the way, that most people don't know about, she tried it out on herself. And she had to wade down through all of these diseases in alphabetical order because they won't rank them. And she gets down, and then her top ranking disease was drooping eyelids. She said, "That's funny, I just had the operation for that 3 weeks ago." so it's sort of just a wonderful confirmation. I mean, I'm not saying it always works, but it just sort of shows that it can be very powerful. And I guess, to answer your question, my point is that lots of people are just voting with their feet and doing this. I think there's a huge demand out there. The thing that everybody talks about Gattaca, the thing that was so sinister about Gattaca was that it was all within a totalitarian environment where it's imposed. And that, of course, I totally abhor. But I think in a democratic, free choice argument, I mean, certainly for adults, go for it. Why not? Now, I'm not going to stray into stuff about kids, but because you get into all sorts of issues there. But I mean, if you look at the eating disorder stuff, I think you'd say, it's the parents' duty of care to their child. I think it's a very good case for the parents having that information about a child and knowing that they might be a very high genetic risk of any of these. Any of these things which has onset before those people become 21 or 18 or whatever it is these days. I think that parents have got a right, got a good case to know.
Yeah.
Anyway, that's for debate. I'm not an ethicist, I'm a scientist.
And I suspect there might be two sorts of people in the world, those who want to know.
There are. There are lots of people who don't want to know
anything.
And the other thing that you can do with those lists is say, I want to know these diseases, but not those ones. So a lot of people don't want to know their Alzheimer's risk, for example.
Yeah.
Which you can understand.
Yeah. Okay. Do you want to, can you shout, or do you want to go to the microphone?
Yes, I should probably go.
Sorry, it's a bit of an obstacle course there, but...
Thanks, Nick. That was fabulous. There will be a number of people who will want to know what the interaction between the environment and the genetic predisposition is. You've already indicated that, for instance, education level is associated with the genetic predisposition. So what are your views about the interface or the interaction between the environment and genetics in the development of disorder?
Well, you've got to be very careful how you use the word interaction. A lot of people use it rather loosely to mean what I would call coaction, that both are working. To a statistician, interaction means that, a gene in this environment will have quite a different effect in that environment. And there are lots of studies attempting to look at that. It's very hard to get that evidence for most traits. There's not really a lot of evidence for that around. I think that in that formal statistical sense, genotype-environment interaction is much less common than people think it is. Most things are just the additive action of genes and environment. Now, I'm not in any way saying that environment's not important. When we look at the heritability, 1 minus the heritability is the environment. But by the way, that also includes measurement error, which as a psychometrician, I'm sure you'll appreciate, it's very large for many of these traits. To the extent that, that there is environment, let me sort of tell you an anecdote which makes the point. So a colleague of mine looked at the heritability of educational attainment in Norway before the Second World War, when there was really very poor access for poor kids getting to higher education. And then after the Second World War, when they had sort of socialistic policies that made everyone able to go, and what they found was that the heritability before the war was about 40%, and then after the war, when things had settled down, it was about 80%. And why is that? Because you've actually removed the environmental variance. You flatten stuff down, and people are actually finding their own level. So I mean. That's partly answers to your question, I think. I mean, yes, you have to take both into account, but you have to think very carefully what you mean by it.
Other questions? I guess the environment becomes pertinent when you're, again, going back to the testing. If you're going to test people, then you need to think about protective environments. Do you think, so if you think your child's going to develop anorexia nervosa, you would have to think about developing protective environments as they grow up, not accessing social media, having regular meals together, those sorts of things.
Well, you're the expert on anorexia. I can't speak to that, but just think about depression. I think if you told someone, or you told a parent about they had a very high risk of depression, then, what do? You insist they get up early and go for a walk, staring into the sun. They go to a gym or play sport. They join activities clubs and so on. They don't sit around their room playing on Game Boys. And you're much stricter about that than you are, with your average child. I mean, there are all sorts of things you can do, to improve mental health. And I think knowing that you're at that genetic risk, I mean. There are lots of people in this room who know far more than I do about how you would do that. But I mean, I think this is really useful information for people involved in behavioural modification.
Yeah.
I mean, I suppose you could argue that that for clinicians, by the time that the clinician gets to see them, the damage is done, and they're already down there. But in terms of prevention, I think this could have some role. And I think particularly for schizophrenia, I mean, I think that we've got such a strong signal there now that I think there's really a case for putting that into use, because in a school, 1 in 100 kids is going to end up with schizophrenia. If you could spot those people really early. I mean, Pat McGorry's always going on about this, but wanting to use behavioural criteria, which may have some role as well. But I mean, I think that actually adding genetic information like this. Now, of course, then the problem becomes, well, in what in what context do you collect that genetic information? I think there's a case to be made for genotyping everyone at birth, actually.
Right, okay. Other questions from the audience?
Well, can I just follow on from that for just a second, because the topic I had to skip was pharmacogenetics, which is actually genetics of drug response. And I mean, a reason for actually having everyone's genetic profile is that where we're heading. And we can partly do, I was going to show you an example, but we already sort of partly predict which people will respond to which drug and which ones will not. Now, at the moment in the medical system, a huge amount of money is wasted. I mean, only about a third of people respond to antidepressants well, and about 20% respond very badly. And if we could predict who those people were and what the better medication would be, you'd be saving a lot of angst for the patients and their clinicians, and you'd be saving a huge amount of health dollars.
Yeah.
Sorry.
Thank you, Nick. Thank you. Mine's a comment really about this whole conversation we've just been having, and that is how it would change the face of research and preventative research when we actually know from the beginning what is preventative, rather than trying to find the associations with it with control groups at this stage when we don't know. We're making assumptions around people's behaviour and behaviour modification to assume that, if you do mind games or have more brain activity, that you might be less likely to develop Alzheimer's. But if you know from the beginning you've got a genetic risk, I think it changes the complexion of the research that we end up doing to get to that actual answer of what are the like you were saying, is it about not looking at fashion magazines for anorexia? We don't really know. It's just something that we've used and it's come out as an association at this stage rather than a cause.
Yes, exactly. Yeah.
And I'm just wondering I'm slightly not pleased about the drug for weight loss, as you can imagine, with my background.
You think they'll be lining up for it?
I think all my healthy weight Yeah, they will be lining
I'm
up.
sure this will be prescription only.
I'm just wondering how long you think it will take to get, what's the timeline here for those?
Well, I think the fastest that they can do it is about, well, we used to say the fastest they could do is about 10 years, but I mean, as we've seen with COVID, I mean, we've got drugs on the market and, in 18 months or something or less, a year. Absolutely remarkable. So it shows it can be done. It's just a question of political will. Yeah, it can be done.
Okay.
But I mean, if you want to tackle the obesity epidemic, nothing else is working.
But what about, because I noticed there were things up on the weight gain side of things. Can we give them a tablet to gain weight? Knock out wasn't there stuff up there?
Some of these are genes that increase weight. So what the drug, I mean, those are also potential targets for the pharmaceutical industry, but not as clear cut as this. I mean, the great thing about this is that those patients are alive and perfectly normal. So that tells you can really kick that gene down without harming the patient. I mean, obviously, lots of trials need to be done of that, but that's the sort of underlying assumption there that makes it potentially a really excellent drug candidate. I don't know enough about this field to know what one would do with that, but I think they're interested in those too. But at the moment, all the efforts going into that one.
Just thinking we could give them a tablet for weight gain while they're on the ward, and I'm sure that.
Oh, exactly. Yeah. Well, you could use those ones for.
I'm sure they'd take it.
patients.
No worries.
You might. Yeah. Absolutely.
No.
General population, I don't think, Tracy.
No. Is there a last question before we close off tonight? Or is everyone thinking they'll rush off and do that genetic testing thing and find out about their drooping eyelids?
It's great fun. Great fun.
Okay, I'd like to thank you very much, Nick, and I'd particularly like to thank Nick because I know that flying around the country, as we know, is not that easy, and it has been a little bit fraught. He did have to do the COVID test, and they took a bit of your brainstem out with it, I guess, and he had to isolate for a little while. But SA Health processed the test very quickly, which was wonderful. So thanks for sharing your knowledge, and particularly for all this new data and helping us to look to the future a little bit, to what's coming. So I'd like to thank you all for coming along, and very much appreciate your support for tonight, and we hope that we can host you again at future ORMA events, and I hope you have a wonderful night tonight.